Hi guys, how are you? This is the part 1 of solving May 2025 paper for Unit 1. And let's get started with the first question. One risk factor for cardiovascular disease is a short blood clotting time. One test for measuring blood clotting time is the prothrombin time, or PT in abbreviation. Read through the following passage about prothrombin and the blood clotting process and complete it by writing the most appropriate word in the dotted lines. Prothrombin is transported in the blood of humans. It's a substance that flows in the blood and it's used when in need. It is converted into thrombin by an enzyme called, let's take a look at the blood clotting cascade here. If we want to convert prothrombin into thrombin, this is done by the enzyme thromboplastin, which is released once a blood vessel is damaged. So my answer is going to be thromboplastin. The substrate for thrombin is fibrinogen. Thrombin is an active enzyme that converts the soluble protein fibrinogen into the insoluble protein fibrin, which is going to form a network of fibers that traps the platelets and blood vessels, forming the platelet plug eventually. Alright, which pair of drugs is used to treat patients who have short blood clotting times? Well, actually, if these patients have short blood clotting times, let's say short PT or prothrombin time, this means that they form a blood clot or a blood scab very quickly, and this causes excessive coagulation. So if I want to reduce this, I'm gonna need to reduce the process of coagulation as a whole. And the two drugs I'm gonna need are gonna be anticoagulants and platelet inhibitors. Platelet inhibitors are gonna inhibit the release of thromboplastin from it, where when the platelets get sticky, they're gonna release thromboplastin in order to allow the platelet plug formation. So my answer here will be it will be B. And why is it B and not A? This is because antihypertensives are not related to this. They only treat high blood pressure. It's not C because statins are for high cholesterol, and of course antihypertensives are for high blood pressure. Platelet inhibitors is going to be correct, but statins is not correct, so my only correct answer is B. Part C, the range for a healthy prothrombin time is between 10 and 13 seconds. A person with a PT of 8.5 seconds was treated with a drug to increase the prothrombin time to 10 seconds. Calculate the percentage increase. Well, actually the percentage increase has this formula of new value minus old value divided by the old value times 100. What is the new value here? It changed from 8.5 to 10. So the new value is 10 minus 8.5 divided by the old value, which was 8.5, all multiplied by 100, which is gonna be 17.6. So just write your answer here, and let's move to the next question. Hemoglobin transports oxygen and carbon dioxide around the body, and we know that because when oxygen binds to hemoglobin, it will form oxyhemoglobin, and when carbon dioxide does too, it forms carbaminohemoglobin. Hemoglobin binds to oxygen in the lungs and releases it in the tissue. Of course, it's released in the tissues when it needs to be used in the aerobic respiration. The percentage saturation of hemoglobin plotted against partial pressure of oxygen is called the dissociation curve. And this is the shape of the dissociation curve, which is already common in our exams, which describes this dissociation curve. Is it a linear curve? Of course not. Linear curves should look like that. So B and A are excluded. It's a sigmoid curve which shows the S shape. This means that it's a sigmoid curve showing a positive correlation as it's increasing in this shape. So my answer will be D. Which name is given to the effect of increasing carbon dioxide concentration on this dissociation curve? It's known as the Bohr effect, where briefly this graph shows that having a low partial pressure of oxygen means higher affinity for the hemoglobin to have oxygen, and the opposite goes for having a high PCO2. It decreases the affinity of oxygen to hemoglobin, which explains why oxygen is released at respiring tissues for respiration, and it's released from the oxyhemoglobin, dissociating, forming only the hemoglobin. Explain how the structure of hemoglobin is related to its function in transporting oxygen and carbon dioxide. Well, first of all, hemoglobin is a quaternary structure protein, which is also globular in shape. Let's take a look at its structure. This is the hemoglobin. It's overall globular, having this ball shape, which makes it soluble in water. So this is one point I'm gonna need to mention. It also has 4 heme groups. These are the heme groups present here, and it has 4 of them, which allows each hemoglobin molecule to carry 4 oxygen at a time, 4 oxygen molecules at a time. We can also mention the part where each one of them has a different affinity, where binding of the first oxygen molecule to hemoglobin has a different affinity for oxygen at different partial pressures of oxygen. We can also give credit to the carbon dioxide part, where it asked me for the carbon dioxide. Carbon dioxide binds to the amino groups of hemoglobin, which is why it forms the carbamino hemoglobin at last. We can also say that this carbamino hemoglobin is formed at low concentrations of oxygen, where the relation is kind of inversely proportional. If the carbamino hemoglobin is formed, this means that we are at low concentrations of oxygen, and when it's dissociated, this means we are at high concentrations of oxygen. So let's write our answer in 2 words. And now let's move to the next question. The structures of blood vessels relate to their functions. The diagram shows part of a cross-section of an artery. Complete the diagram to show details of the structure of an artery and label 2 features of an artery on your diagram. We only got here the lumen and another circular part surrounding it. We don't have any detailed parts and labels, so let's add them to the diagram. Let's say that this outermost part is the tunica externa, and we can draw a circle just next to it, and this will be the tunica media, just in the middle. Next, we can say we have a basement membrane surrounding the tunica intima, and the tunica intima is the innermost part which has the lumen inside it. The table shows some dimensions of 2 veins, the vena cava and a small vein, which is the ratio of the diameter of the lumen to the thickness of wall in the vena cava. The diameter of the lumen is 30 millimeters and the thickness of wall is 500, but note the differences in units. This is in millimeters and this is in micrometers. So I have to convert one of them to the other in order to have an accurate ratio. If I want the ratio of the diameter of the lumen, I'm going to start with the lumen, which is 30 millimeters Let's convert the 30 millimeters into micrometers, and we're gonna multiply by 1000, making it 30,000 to 500. You can simplify it using your calculators, and the simplest form will be 60 to 1, which is D, so D is my answer. The next question was kind of tricky, so pay attention to this idea. Calculate the cross-sectional area of the lumen of this small vein. Use the formula cross-sectional area equals pi r squared, which we already know. Give your answer as a whole number. Take care of these notes written at the end of the calculation questions, whether they need you guys to round it up to a number of decimal places or significant figures, writing it as a whole number or not. This will make you lose marks if you didn't do that. Alright, let's get back to the question. We need the cross-sectional area of the lumen of this small vein. So if I need the cross-sectional area of the lumen, this means that I have to get the diameter and the radius of this lumen first. Do I have it in the table? No, I don't have it. So how am I gonna get it while I have the total diameter of the vein and the thickness of the wall? So let's actually imagine this. Think of it from the diagram here. We know this is an artery, but it has the same idea. If the total diameter of this whole thing is 46 micrometers, and we have the wall of it, the wall surrounding it is in two parts. So I have a wall here and a wall here. So the wall is times 2. We're gonna recap on this idea now too, as well. Let's say we have this vein. The total diameter of this entire vein is 46 micrometers, but the wall here is found 2 times, once in the left side and once on the right side. So it's gonna be 4 on the left side and 4 micrometers on the right side. What am I gonna do if I need only the lumen here? Of course, I'm gonna remove the part of the wall. How am I gonna do this to calculate the diameter of the lumen? We're gonna do 46, which is the total diameter of the whole vein, minus 2 times 4, which is 8, and this gives me the entire diameter of the lumen only. So it will be 38 micrometers. Now I got the diameter. This is the diameter of the lumen. Can I use this? No. We need the radius to substitute into the equation. So the radius simply is the diameter divided by 2. 38 divided by 2 is 19 micrometers. Now we can finally use this to get the area. Area, or the cross-sectional area of this lumen, is pi times 19 power 2. Use your calculator to get the answer, and don't forget to round it up to a whole number. The answer will be 1134.1149 and so on. I only need this answer, so just write it here. Don't forget to write it in the spaces provided to get the answer. And that's it for part 1. Thank you guys for watching. I hope you found this useful.